Assalamu alaikum students. I am Fauzia Iqbal from Multan Public School and College. As you know, I am your biology teacher. Today I will explain you the remaining topics of chapter number 5. So the first topic is significance of meiosis. In significance of meiosis, you are told that it was first of all described in 1890 by a German biologist August Weismann. He pointed out that meiosis was necessary not to maintain the number of chromosomes in the next generation but also to produce variations in the next generation. So I will explain you these 2 points. Number 1 point is maintenance of chromosome number in the next generation. Meiosis is essential for sexual reproduction. So first of all, I will tell you about humans. In humans, diploid gamete mother cells or germ-line cells undergo meiosis to produce haploid gametes. So these gamete mother cells are diploid and they are to produce gametes. So they will undergo meiosis and they would produce haploid gametes. Male gametes and female gametes. These two would unite to form diploid zygote. And then this diploid zygote undergoes repeated mitosis and it develops into next diploid human. So this is all about sexual reproduction in humans. Now I would tell you that what is happening in case of fungi and protozoan. If fungi and protozoan are already haploid, as in case of many haploid fungi and protozoan, they produce haploid gametes through mitosis. Because they are already haploid, so they produce their gametes which are haploid by mitosis. Now I will tell you about plant's life cycle. As it is shown you here in this diagram also. You can see that I have shown you alternation of generation. In plant's life cycle, 2 generations are alternating with each other. One generation is known as sporophyte generation because it produces spores and it is diploid. So this sporophyte generation produces haploid spore by meiosis, which grows into haploid gametophyte. Spores grow into haploid gametophyte generation. And why gametophyte generation is known as so? Because it produces gametes. So gametophyte generation produces haploid gametes through mitosis. Okay, by mitosis, not by meiosis, because meiosis has already taken place at the time of spore formation. So now the gametes are being produced by mitosis and these gametes would combine to produce diploid zygote. And then zygote undergoes repeated mitosis to produce diploid sporophyte. So this is all about the maintenance of chromosome number in the next generation in humans, in fungi, protozoan and in plants. Now the second point of significance is production of variation in the next generation. The chromosome pairs of each parent undergoes crossing over during meiosis. So daughter cells, that is gametes, have genetic variation. Okay, due to crossing over, variations are being produced in gametes, and when these gametes would fuse with each other to form zygote, in the next generation, the new one would be having different genetic makeup with respect to parents. And thus meiosis allows a species to bring variation in the next generation. And these variations are known as beneficial variation because due to these variation, the organisms can withstand the changing environment. Next is errors in meiosis. As you know, what is happening at anaphase 1 and anaphase 2. At anaphase 1, chromosomes separate and go to the opposite poles, while during anaphase 2, sister chromosomes separate, and all this process is called disjunction. This is a normal process. but sometimes the separation of these chromosomes is not normal and it is called non-disjunction. So this is the error. This non-disjunction is the error in meiosis. It results in the production of gametes which have either more or less than the normal number of chromosomes. As you should know that number of chromosome is fixed for each species, but as a result of this non-disjunction, number of chromosome can alter in the new one. As you know that if such abnormal gamete fuse with normal gamete, it will result into abnormal chromosome number in the next generation. Here you are given the example from humans. As you know that in humans the normal number of chromosome is 46, but due to this non-disjunction it can result into 47 or 45 number of chromosome in human, and it would cause some abnormality in humans. Now the next topic is comparison between mitosis and meiosis. As you know that meiosis 2 is similar to mitosis, while meiosis 1 makes the actual difference between these two types of cell divisions. Here you are shown this diagram to compare mitosis and meiosis. As you know, at prophase of mitosis and meiosis, chromosomes are different from each other at these two steps. As homologous chromosomes in case of mitosis do not form pairs and there is no crossing over, while in prophase 1 pairing of chromosomes occur and crossing over between the homologous chromosomes takes place. Now look at the metaphase. Metaphase of mitosis and meiosis 1 is different again. In case of mitosis, single chromosomes align to form metaphase plate, while at meiosis 1 homologous pairs align to form metaphase plate. Here you can see that homologous chromosomes are present in the form of pairs and they are aligning on the metaphase plate in parallel way. Now look at the anaphase. Anaphase of mitosis, you can see that chromosomes break and individual chromatids are being pulled towards the poles, while in case of anaphase 1 of meiosis, individual chromosomes of these two pairs are being pulled towards the poles. What is happening at the telophase? That at the end we can see that at the end of mitosis two daughter cells are produced, and these two daughter cells are having same number of chromosomes as were present in the parent cell, while on the other hand in case of meiosis, when meiosis 2 will take place, at the end there would be 4 daughter cells, and these 4 daughter cells are haploid with respect to the parent cell. So this is all about comparison between mitosis and meiosis. When you will draw this diagram, you will better understand the difference. Now I will tell you about apoptosis and necrosis. Apoptosis and necrosis are two programmed cell deaths. In case of apoptosis, the main apoptosis is one of the main type of programmed cell death, means it is already programmed. During apoptosis, cell shrinks and become rounded due to breakdown of cytoskeleton by enzyme. Its chromatin undergoes condensation and nuclear envelope breaks. In this way, nucleus spread in the form of small units, chromatin bodies in the cytoplasm. Cell membrane makes irregular buds. As you know, buds are the outgrowth, so these irregular buds here are known as blebs. These blebs break off from the cell and are now called apoptotic bodies, which are then phagocytosed by other cells. These apoptotic bodies would be eaten up by the other cells, means those would finish from that place. You can see all the stages or sequence of this process of apoptosis. At first stage, you can see shrinkage of cytoplasm and disintegration of nucleus, and then blebs production, and then apoptotic bodies are being formed. Same process can be seen in this diagram also, that shrinkage of cytoplasm and nuclear material, chromatin material is broken down, and blebs are being formed, and blebs are being broken from each other, and then apoptotic bodies are being formed, which would be phagocytosed by the enzymes of the other cells. Now I will tell you about the significance of apoptosis, that what is its importance. Apoptosis can occur when a cell damage or undergoes stress condition. Apoptosis removes the damaged cell, preventing it from getting further nutrients or to prevent the spread of infection. Okay, so this is its significance. It is good for the place where it is taking place. You are told another significance also, that apoptosis also give advantages during development. For example, during the formation of our fingers, when a new one is being developed, when its fingers or other body parts are to be formed, the cell between them undergo apoptosis and the digits separate. This is example from the formation of fingers, and same is the case for the formation of all body parts of an organism. As body parts are to be made, those are shaped by apoptosis. Now I will tell you about necrosis. Necrosis is the accidental death of cells and living tissues also. Necrosis is less sequential than apoptosis, means as many steps we have gone through in apoptosis, those many steps are not taking place in necrosis. There are many causes of necrosis, like injury, infection, cancer, etc. Necrosis may also occur when a cell is given a hypoxic condition, a hypoxic environment, means if some area or some cells of the body are given less oxygen availability, then those cells may cause death. During necrosis, there is release of special enzymes from lysosome. Lysosomal enzymes break cellular components and may also be released outside of the cell to break surrounding cells. Cells that die by necrosis may also release harmful chemicals, and due to those harmful chemicals, other cells may also damage. We can take the example of spider bite or some other insect bite, which may cause necrosis in some area. Okay, and similarly necrosis may be due to lack of proper care to the wound site. If there is some wound, some injury, and some proper medicine is not taken, proper care is not taken, then necrosis may occur in that area. This is the end of this chapter, dear students. Now I will tell you about homework, homework related with these topics, today's topics. I have given you here some short questions. Number 1 short question is, how does meiosis produce variation in next generation? Number 2, describe non-disjunction in meiosis. Number 3, define apoptosis and necrosis. Number 4, plants don't make their gametes by meiosis. How is that? Number 5, draw comparison between mitosis and meiosis. Hope you understood all this topic, and as you know, this is the end of this chapter. Now you will prepare its test also. I will take your test and I will tell you about its schedule also. And this is time to say goodbye. Okay, Allah Hafiz.